Everyone is talking about GLP-1 drugs, Ozempic, Wegovy, Mounjaro.
Maybe you've heard the jingle. Or maybe you've seen Ozempic in the news recently.
Weight loss.
Weight loss.
Easy weight loss.
Miracle diet drug.
Miracle weight loss drug.
This is a game changer.
Because the popularity of these drugs have exploded, Novo Nordisk has increased production, making millions of pens in facilities like this one that are operating 24 hours a day, 7 days a week.
But that's still not enough.
It's a whole new class of drugs that treat diabetes, drive weight loss, silence hunger, curb addictions while protecting the heart, the kidney, the liver, and they may even extend lifespan.
These medicines don't just suppress hunger. We now know that they reach into the oldest circuitry in the brain, the system that generates desire, and they turn down the volume.
Grocery bills are falling, alcohol sales are going down, and even casino revenues are declining.
In fact, airlines are recalculating fuel loads for lighter passengers, and the junk food industry is alarmed.
I bet. And now experts are asking a question: what happens to an economy built entirely on desire when desire itself goes quiet?
This is something no medicine in history has ever done.
Join us in the next few episodes as we journey from surgical wards where diabetes disappeared virtually overnight to the ugliest fish on the planet, and from smelly labs to a lizard in the Arizona desert.
Welcome to the Discovery Dialogues podcast, where we take a deep dive into the history of groundbreaking discoveries in the life sciences. I'm your host, Ani Deshpande. Each episode, we will uncover some incredible, insightful, and often just insane stories behind breakthroughs in medicine. Join us as we journey through the twists and turns of scientific history, one breakthrough at a time.
I'm Pam Itkin-Ansari, a diabetes and pancreatic cancer scientist at Sanford Burnham Prebys Institute in California.
And I'm Ani Deshpande, a cancer researcher also at Sanford Burnham Prebys. Welcome to episode 7 of the Discovery Dialogues podcast. In this episode, we are delighted to be joined by some of the scientific luminaries who made these drugs possible. Starting with Jens Juul Holst, a pioneering physiologist and surgeon who helped uncover GLP-1, Svetlana Mojsov, a brilliant biochemist whose work revealed its active form, and David Nathan, one of the world's leading specialists on clinical trials in diabetes, who helped bring these drugs to the clinic. And even though we are just using excerpts of our conversations with them in this episode, we will release the full conversations as Discovery Dialogues deep dives.
Let's start with an observation that really puzzled surgeons in the 1970s as obesity was becoming a major health problem, and surgeons were performing a new operation called gastric bypass. This surgery shrinks the stomach and reroutes part of the intestine, so patients can't eat as much. The expectation was simple. Patients would gradually lose weight over the next year, and if they had diabetes, doctors assumed it would improve slowly as the weight came off. But the doctors were in for a big surprise.
Yes, and in fact, in many diabetic patients, within 2 or 3 days of the operation, their blood sugar returned to normal, no longer requiring medications. And this was long before they had lost any meaningful weight. It was as if someone had simply switched the disease off.
And this is exactly the question that intrigued Danish physiologist and surgeon named Jens Juul Holst. Holst realized that gastric surgeries were leading to increased insulin. So what was going on here? Why were surgeries of the gut increasing insulin release from the pancreas?
Here's Professor Holst from Copenhagen, Denmark.
We have hormones that are secreted in the gut. And what they do is to stimulate insulin secretion, and they're secreted when you eat. So they're being stimulated by food intake, nutrients. And it turns out that to be able to secrete sufficient amounts of insulin, we need these hormones. It's really, really important. And if you don't have them, then you get glucose intolerance, then you get diabetes.
You know, Pam, this is quite mind-blowing actually. I've always thought that you would get diabetes from a problem in the pancreas, but what he's talking about is that the lack of hormones made in the gut can lead to diabetes. And maybe what was happening in the bariatric surgery patients was that some of these gut hormones that had yet to be discovered were rising, and that's why their diabetes went away.
So the theory came up very quickly that perhaps we were looking at an effect of insulin-releasing hormones from the gut. So that became my kind of destiny in science.
Wow, so there are insulin-stimulating hormones in the gut?
Isn't that cool? And that raised an even bigger possibility. If these gut hormones could be identified, they might become powerful new medicines.
You know what I find really fascinating is that these gastric surgeries had not even touched the pancreas where insulin is made, but what they had perhaps done is to rewire the communication between the gut and the pancreas. And this was called the incretin effect. As Svetlana Mojsov explains,
It was a clinical study, very small one. It was shown that if glucose is taken orally by mouth, there was a higher levels of insulin compared when it was administered intravenously. And that immediately suggested there must be peptides, which are secreted in the intestines when we eat a meal.
Isn't that amazing that sugar taken by mouth triggers much more insulin than the exact same sugar given straight into the bloodstream? Wouldn't you have thought it could be the opposite? But actually, it does make sense because the food we eat has to first go to the gut, not directly into the bloodstream.
exactly. And here's Dr. David Nathan.
So it looked like this was something that was actually a natural signal from the gut. Food goes into the gut. It says, hey, food's coming. You better start making insulin. This is the chemical that brings the message to the pancreas. Start making insulin because there's food coming in any minute.
Then it became clear immediately that incretins can be used as new medicines to treat diabetes. And that's why there was a really big effort starting in the 1970s to discover the incretins.
To find the incretin responsible for the diabetes reversal, Holst and other scientists frantically looked for hormones in the gut, and they found one that looked a lot like the hormone glucagon that is made in the pancreas.
And so, to find what was now called glucagon-like peptide, they'd have to wait until they found out more about glucagon.
Now, the real glucagon is, in a sense, insulin's mirror image in the pancreas. If insulin lowers blood sugar, glucagon raises it.
Right. Think of it like a thermostat. The two hormones work in opposition, constantly adjusting the blood glucose so it never gets too high or drops too low.
Some scientists wondered whether the thermostat was broken, that in fact, diabetes was not just a problem of too little insulin activity, but also of too much glucagon.
This was the 70s, and it became obvious that glucagon plays really a critical role in controlling glucose metabolism. And so in our thinking in Marifu's lab, we thought that, well, it's possible that if we develop an inhibitor of glucagon, something which will suppress glucose, and it will be administered together with insulin, we will come up with a new and better treatment for diabetes. So that was the motivating force.
So this was an elegant idea. What if managing diabetes wasn't just about insulin, but also about carefully adjusting glucagon to restore balance between the two?
But first, scientists had to know how glucagon was made. The gene for glucagon had not been cloned. This was the 1970s, and there was a big problem.
Yes. As we described in episode 2, fearing misuse of gene cloning technologies, there was a ban.
A new technology had evolved called recombinant DNA technology. There was a concern that the artificially made DNA could get into the sewer system and become a hazard to people.
This is the recorded voice of Dr. Joel Habener, a preeminent Harvard physician scientist whose work propelled the field forward.
As we repeatedly show in our episodes, when scientists are faced with hurdles like these, they often look at how nature has solved some of these problems.
And even from the most obscure creatures, you can get insights that profoundly advance medicine. And this is the kind of inspiration that hit Joel Habener from the ugliest fish in the ocean.
First, Habener realized that the ban on cloning only applied to warm-blooded animals, but not to fish. And so he turned to a member of his lab who knew a guy.
Who knew a guy.
Yes, this guy was a fisherman who routinely supplied labs in Boston with fish for their experiments. And the fish he chose for this research was the anglerfish, this deep sea nightmare with bioluminescent lure dangling from its forehead, the gaping jaws and the needle-like rows of teeth. It's not an animal that inspires affection. But besides being cold-blooded, the fish solved another problem.
Exactly. As we discussed in episode 1, the trouble Banting had purifying insulin was because the pancreas of mammals contains 2 compartments. One part makes hormones, but the other part makes digestive enzymes powerful enough to chew up biological molecules. When you grind the whole thing together, your precious hormone messages can get destroyed in the mess.
Exactly. And this is really where Habener's approach was so elegant. In the anglerfish that they had caught off the coast of Cape Cod, the compartment that makes digestive enzymes is conveniently separated from the one that makes hormones. In fact, you can even see it with your naked eyes. And for a scientist who was trying to study hormones, this was pure gold.
Oh, wait. There's another reason scientists use this fish, because no one else wanted them. As Habener himself said in an interview to the Harvard Gazette, it's a trash fish. It's big and it's ugly. You throw them back into the ocean. And if you're watching this episode on YouTube, you'll agree. The anglerfish is one ugly animal.
Okay. And so from this fish, Habener's group finally found the gene for glucagon. And here there was a big revelation. And this was informed in part by the important work of geneticist Graham Bell. Besides glucagon, this gene could also make tiny glucagon-like peptides. And this immediately energized the field because were these glucagon-like peptides the missing incretins?
And then came the moment that should have solved everything. They had the peptide. They made it in the lab. And then they tested it.
So Svetlana looked at the structure, and so did we. We synthesized it, and so did Svetlana. And I put it on my pancreas, and it didn't work. It didn't work at all.
And it wasn't just the Danish and the Boston groups. Others tried it as well, and nothing happened.
So all this work for nothing? Ani, at this point, one might rethink their career. But they didn't. They kept going. To get a definitive answer to this question, they performed the arduous task of making new tools, antibodies, that could detect all the different forms of peptides from the glucagon gene. And Holst went straight to the source, the gut itself.
That's why we, realized that we had to pull out the real peptide ourselves from the gut, and that's what we did. We went the classical way of isolating the peptides from the body, the natural peptides. So no speculation, we simply went for the molecule itself.
At the same time, Svetlana Mojsov, who was now closely working with Joel Habener in Boston, had an epiphany.
I was really deeply involved in glucagon, biology, biochemistry. I knew its sequence backwards and forward. I was trying to see where there is a similarity to the glucagon sequence. And you can also see here how I was counting where the active sequence starts. So this was really my notes. That's where I predicted that it's going to start at histidine and it's going to be the biologically active peptide. So it's very clear. So this is it.
In other words, Mojsov realized that for the peptide to work, a tiny piece had to be chopped off.
Ani, given the importance of this discovery, I really had goosebumps when I saw Mojsov's original laboratory notebook, the raw data showing the sequence with markings in her own handwriting.
Yes. If you're watching this on YouTube, you can see this on the screen. Even though she made it sound simple, it wasn't. Mojsov, working in Boston, made the shorter active peptide and was almost single-handedly raising antibodies in rabbits. It was so distasteful that every time she did that, she had to just go home and take a shower. Jens Holst, the Danish surgeon, on the other hand, who was purifying the active peptide from mashed up intestines, he also had fond memories.
There was a certain smell in the laboratory those days when we were boiling gut extracts, and things like that. So there was a lot of that also.
Oh, the glamour of science.
Exactly. But all this hard work paid off. In Boston, Daniel Drucker, then a young Canadian trainee in the Habener lab, showed that this tiny peptide could stimulate insulin production in cells grown in the lab. Drucker made another really big observation. He showed that GLP-1 stimulated insulin production only when high levels of sugar were present.
Right.
It was like a smoke detector that only goes off when there's an actual fire.
So this wasn't just any hormone. It was the gut's food sensor telling the pancreas that food is coming, start making insulin.
Exactly.
Now they wanted to find out if this glucagon-like peptide could stimulate insulin release from the whole pancreas of an animal. But to do this, they needed an almost unbelievable setup. A pancreas kept alive outside the body with fluid pumped through it, so it would still behave like a working organ.
Which is not easy.
Not easy at all. Well, in Denmark, Jens Holst was a highly skilled surgeon who had done just this with the pancreas of the pig. In Boston, Mojsov worked with Gordon Weir, who had developed a system to keep a rat pancreas alive outside the body. And then the results came in.
It was a double-blind study. We'll collect samples, we will divide them in half. Half of them will stay at the Jocelyn, and they will analyze it for insulin, and half of them I will take it to the Mass General, and I'm going to analyze it for GLP-1. And we said that we are not going to exchange it until we had the data. And I sent the data, and before I could even realize, Gordon Weir called me on the phone. And he said, "You won't believe how active it is." He says, "I've never seen anything like that."
And meanwhile, in Denmark,
pulled it out of the extracts and purified it and put it on my pancreas again. And we could see that it did all that it was supposed to do.
So here was the missing proof.
Holst also found an answer to a puzzle that had been bothering him for years. He showed that GLP-1 levels in the gut rise in response to food, and that GLP-1 rose even more dramatically after bariatric surgery.
So that explains why their diabetes could almost disappear overnight. Now, the golden question was, would this molecule do in humans what it did in the lab?
I think it was Gordon Weir who said, well, now we have to go into clinical studies. You have to put it in patients.
At the Mass General Hospital in Boston, David Nathan was working just down the hallway from Joel Habener and Svetlana Mojsov.
So he came down the hallway and Joel said, look, we have these new data. And he said, what do you think? I said, wow, this looks like the Incretin that everyone's been looking for. So we turned our attention to looking at the GLP in humans. They purified the stuff. We got the FDA to clear it, put it into intravenous bags, and I designed these studies to give it to humans, both humans with diabetes and humans without diabetes, to see whether they make insulin, what happens to their sugar levels.
What Nathan described to us was a classic clinical research unit in the late 1980s. Research nurses drawing blood every few minutes, patients fed and monitored, bags with GLP-1 going into an IV line, and importantly, nobody knowing which person was getting the real molecule and who was getting the placebo.
And while Mojsov and her colleagues were working with the blood samples in the lab, David Nathan was watching how these patients responded in the clinic. We asked both of them about that day, and here's what they remember.
Again, double-blinded study, but the way we arranged them in the gamma counter, we will just put insulin and then GLP-1, and it was clear every single individual responded. It was just incredible because usually in clinical studies, you don't expect every individual to respond.
So we dripped this stuff into these patients. Some had diabetes, some didn't. They didn't turn blue or green, or they were fine. And we're measuring frequent blood. Our research nurses are drawing bloods out of them. We're feeding them. We're dripping glucose. We're measuring this. And sure enough, when the results came back from the laboratory, insulin goes skyrocketing up. And their glucose levels, even after we fed them, didn't increase. They stayed stable or decreased, showing that this GLP, this is the first demonstration in a person with diabetes that the GLPs worked.
Wow. Now, Pam, when I read the landmark paper describing this discovery, there is a sentence at the very end of this paper, and I cannot believe what it says.
As you guys mentioned in the first paper that I wrote on this, I said, whether it goes on to become a drug, we'll have to see. That was the understatement of the century, probably.
Right, possibly of the century, yes.
Yeah.
So, Pam, we now have a blockbuster drug, right?
Well, you could infuse GLP-1 continuously and it would work beautifully. But the moment you stopped, the effect would vanish. And of course, a drug that needs a continuous IV drip cannot help hundreds of millions of people.
So how did we go from something that disappears almost instantly to drugs that are now transforming millions of lives and shaking up society?
Well, join us in our next episode to hear how a venomous lizard from the Arizona desert had solved this engineering challenge through millions of years of evolution.
Thank you for joining us on the Discovery Dialogues podcast.